Hello, my name is Carlina Schubert. I'm a senior here at UHD. My major is biology with a concentration in environmental science. The subject of my research is relationships among functional leaf traits in multiple grass species accounting for phylogenetic relatedness. My PI is Dr. Michael Tobin. His lab concentrates on functional traits that are related to drought conditions and within grasses. And I'm going to get into each of those things. This here is a picture of our STB building with a foreground of sorghum halepense, which is one of the grass species in my data set. So why functional traits? So functional traits are traits related to growth, reproduction, and survival. They're cheap and they're pretty easy to collect, which is great for undergraduate research. And they focus on the phenotype rather than on the genotype. So why grasses? So again, they're pretty easy to collect because they grow everywhere. They're very tough, and so they can survive in our city. They're a low hazard organism to use, so you don't have to worry about your undergraduates getting bit or having like an altercation with them like you might with like wild animals. You can do comparisons within individuals, within species, or cross species. Since these grasses have a tendency to spread out, you can actually sample the same individual many times, or if you space your samples out, you can do several different members of the same species, or since there's a lot of variety of grasses out there, you can do comparisons between species as well. They also share functional traits with some really important agricultural species, such as corn, rice, and wheat. So why drought traits? So climate change is predicted to make the weather more extreme, both wetter and drier. So we don't really yet understand fully how our plants and our agricultural species survive in these extremes. So the more that we know about that in general, the more that we can apply that to our own agriculture. The species ranges where they can survive is often defined by those extremes that they can survive. So if we can better understand how they survive those extremes, then we can better help our own selves. And so, more understanding is helpful. So what's in the data? So these functional traits were collected by Cure Ecology class here at UHD. And the ones that we picked were ones that could possibly be related to drought. We have minimum leaf conductance, leaf mass per area / specific leaf area, those are the inverse of each other. Stomatal density, guard cell length, and recently we started taking widths as well. Leaf width and leaf width. These organisms are all grasses. Some of them are more closely related than others, and that matters for our data set because we need to take that into account when we're making comparisons. So here's the phylogenetic tree for the species in our data set. There's 13 grass species, all of them from the Houston area. We use the phylogenetic least squares method that takes into account the phylogenetic tree. The pared down tree that you see here was made in R using PhyloMaker with PhytoPhylo from Jan and Kang 2019. Source plant phylogeny is an updated version from Zane et al. 2016. And the PGLS code is from Natalie Cooper's Practical Exercises for Methods in Macroecology and Macroevolution Model. Now these top 10 species I have here are my C4s, and these bottom 3 are my C3s. They have slightly different methods for surviving drought, so we wanted to make sure that they are not those differences aren't making some weird skew in our data, so we wanted to keep track of where they are. So the first functional trait that we're going to look at is minimum leaf conductance. So this is an index for the rate of water loss when the stomata are closed. It's measured by weighing drying leaves every 15 minutes for 2 hours. It takes into account the humidity, temperature, and atmospheric pressure on the rate of water loss. And a low G-min would help the plant survive the drought for longer because it's losing water at a slower rate. So here's a picture of a leaf. This is the sorghum halepense, same from the very first slide. I've got a zoom in on, this is an image taken from an impression of sorghum halepense, and these are the stomata. And this is sort of just an illustration of like the water escaping. And so when during the minimum leaf conductance, which is sort of when it's losing water when the stomata are closed, it can either lose the water through the waxy covering that's over the entire leaf, or it can actually lose water through like an imperfect closing of the stomata, and the GMIN measures that total loss. Another functional trait that we'll be looking at is stomatal density. So the stomata are the pores in the leaf that control the gas exchange that powers photosynthesis. And stomatal density measures the average number of stomata per millimeter of leaf. It's measured by making transparent leaf impressions and taking counts under a microscope. And here's an example. We've got a bar for 250 micrometers, and these little hamburger-shaped guys are the stomata that we're interested in. And our students count those under a microscope. And what we want to know is, does the density affect the minimum leaf conductance? So if there's more stomata, more holes in the leaf, does that result in an increased loss of water or a decreased loss of water? But after PGLS, GMIN and stomatal density did not correlate. So we ended up with this sea of dots here. Our squares are our C3s, and the circles are our C4s. And the C3s are not clustering, so we're not worried about them skewing our data. So then after that, we decided to look at guard cell length, thinking that, oh, maybe there's something going on with the guard cell length that's messing with the stomatal density measure. Because if there's some sort of relationship between how large the stomata are and how many a leaf has, that might cause that chaos that we saw right there. So the guard cell length is an average length of the guard cells that control the opening and the closing of the stomata. And this gives us a ballpark measurement for how big the stomata are. It's measured by taking micrographs of leaf impressions and measuring their lengths using image J. Here's an example of some large and small stomata. Cynodon dactylon has very small, and Poa annua has very large stomata. And you can see they have like a pretty different shape to them even. The Poa annua are much longer, whereas the Cynodon dactylon are more circular, which is why we started taking stomatal widths recently, because we thought maybe the longness versus the shortness might have a factor in there. But again, we did not, after the PGLS correction, we did not find that there was a correlation here. And we also tried it against just guard cell length alone, but also did not find a correlation. Leaf mass per area. So we're still looking for traits related to minimum leaf conductance. We looked at traits related to carbon investment in the leaf. Only one available in our data set is leaf mass per area. And this sort of gives you an idea of how thick the leaf is. This is measured by taking areas of the leaf scans of fresh leaves using image J. And these areas are taken during the GMIN measurement, so we already have that information, so it's easy to get the leaf mass per area once we take the GMIN of a species. Then we weigh the oven-dried leaf masses. If there's like a large, our prediction here is that a large investment in leaves would benefit from a low leaf conductance, because you want those leaves to survive drought, especially if you've invested a lot of energy and carbon into them. And we actually did find a correlation here with a p-value of 0.01. And this finally gives us a direction for further study for our cure classes. And we actually got like a negative correlation here. So the GMIN goes down as the leaf mass per area goes up, which is what we predicted. For my conclusion, so we expected minimum leaf conductance to increase with increasing stomatal density, but we did not find that. We tried predicting minimum leaf conductance using our other stomatal measures in the data set, but could not find a correlation. We tried an index for leaf investment, leaf mass per area, and did find a correlation. And we think that this could be a result of a couple different things that we'll have to investigate, but one of the things could be a cuticle thickness. And here are my acknowledgements. Thank you, everybody.